Hey, everybody. My name is Darla White, and I am with you virtually here today to give you a little bit of an insight into possibilities as you pursue a career in marine science. Hopefully, that's what you would like to do because we need more great people out there. And uh, I've been asked to share a little bit of my story because I was a MOP student just like you. Um, University of Hawaii and MOP and Quest and the diving program were all just really pivotal parts of how I ended up in this field as a working professional. And so I'm going to share some of that with you today. And hopefully this is going to go well. Oh, you're moving stuff too? Okay. All right. I'm sharing. Sorry about technical difficulties here. All right. Ta-da. There we go. Hopefully you're seeing the correct screen. um, anyway, so again, my name is Darla White, and this is how I became a marine scientist in Hawaii. I've spent most of my time underwater for the better part of the last 18 years. Um, and just a brief outline of what I'm going to cover today, a brief introduction to me, um, how I got there, other MOP graduates that I work with. Quite frankly, I've worked with most of the people I went to school with at UH Hilo and in MOP. It's Hawaii's good like that. Talk about a few of the opportunities I've had along the way. Um, uh, there are lots of opportunities here in Hawaii. I mean, a lot of them, and um, especially in grad school, you'll have doors fly open in every direction. People just, it's so amazing here. I do hope that you pursue graduate school. Um, I'll talk a little bit about what I did at DAR, which is the Division of Aquatic Resources, which is a division under the Department of Land and Natural Resources. And then I'll talk about some partner projects. So what I mean by that is working with other agencies and organizations such as NOAA or USGS or different universities. And then what's next? I'll tell you what I'm doing today. So I spent a lot of years at the Division of Aquatic Resources. Most recently, I was a Marine Protected Species Coordinator on the False Killer Whale Project. But for 10 years, I was a Special Projects Coordinator here on Maui with the Kahikili Herbivore Fisheries Management Area as one of my primary projects. I've focused a lot of effort and science on coral disease and bleaching. One of our main roles is long-term monitoring, so looking at fish and habitat through time to look at change over time, and then working with partner agencies and NGOs, focusing on question-driven research, which is different from monitoring. Monitoring is looking at change over time, but why that's happening could be a variety of different things. Question different research just asks a specific question, and they're usually shorter projects, like a year to 2 years, maybe 3 at the most. And my personal objective is really where my heart is furthering our understanding of the environment through research and education to promote the conservation and longevity of these ecosystems. Also, the Eyes of the Reef co-coordinator for Maui, along with Donna Brown. And she's going to tell you a lot more about Eyes of the Reef. I'll just touch on it today, but she'll give you the full thing. So this is how I usually look for work. This was my uniform for better part of 18 years. And how did I get there? Well, I actually came to UH Hilo in 1999 and did Quest. And then I got my MOP certificate in biotoxin research. I studied Ciguatera, and that's a whole other talk. Um, when I graduated with my undergrad in marine science, I was hired on as a research assistant to a professor at UH Hilo, where we did more biotoxin research. And then I continued that research into my master's thesis in the Tropical Conservation Biology and Environmental Science program at UH-Hilo, where I got my master's. Um, in doing so, I was able to go on NOAA cruises. So I was able to collect my samples and data throughout the whole of the archipelago and also collect fish data for NOAA, which was a whole lot of fun. And most recently, I have a very strong interest in analytical mapping and spatial ecology. So I left my position at DAR to get more education. I went to Oregon State University and got a master's certificate in GIS, or Geographic Information Science. But it all started with Quest. And I'm sure Donna's talked to you a lot about Quest, but if you are even remotely interested in diving, please do this. Do it sooner than later, because you'll be so happy you did. The opportunities are many, and it's just a world of fun. It's the best day at the office ever. Go take Quest. Just do it. Do whatever it takes to do it. I know there's a lot of grants and things available as well, but talk to Donna about that. But that's where the door opened. From Quest, I was hired onto the West Hawaii Aquarium Project. So this is a long-term monitoring project in Kona, where they've been looking at the effectiveness of what they call the FRAs, which are fish replenishment areas, which were um, basically the primary management tool for the aquarium trade in West Hawaii. So I worked on that project for 7 years. It was amazing. And UH-Hilo also offers an undergraduate thesis. So it's like a master's thesis sort of scenario, but before you go to grad school. And very few colleges offer this. It's a real step up in your ability to to go to grad school. So I looked at Ciguatera on the island of Hawaii, and I compared windward versus leeward shores, and it was a phenomenal opportunity, and I had no idea how valuable it was until after I did it. Um, but it's a great way to get some good research training. Um, and the research project that I was hired to after I graduated was called an assessment of anthropogenic impacts on 2 coral reef sites on the Big Island, Honokohau Bay and Kealakekua Bay in West Hawaii. This was funded by the EPA and the first marine project in Hawaii funded by the EPA. These 2 bays over in West Hawaii have historical data on um, the coral reefs there and the nitrogen inputs into these areas, and they're both slated for development, a lot of development over the next 20 years. So we went in to look at change over time and found some really interesting impacts. But we went out to these 2 bays every month for about 2 years, working with the tides and water quality and coral reef and fish, and it was phenomenal. And all of my divers were students at UH, and we had just an amazing time on this. This work has been cited many times throughout the years. I'm very proud of that work. It was fun. Um, for my master's thesis, we I wanted to look at ciguatoxin or ciguatera. Basically, it's the #1 food poisoning in the tropical regions of the globe. Um, it's it impacts basically fishermen more than anybody, because it's a toxin found in fishes that comes from the environment. It's a single cell algae that produces a toxin, and it moves through the food web. And so it's my contribution to artisanal fisheries science. And this provided an opportunity to look at fish throughout the whole archipelago. So I was invited out on the NOAA cruises, and we went to all the islands and collected fish and fish data. Worked with a lot of different researchers, sharing samples. It was just an incredible opportunity. I went up there 3 different times, worked with all kinds of different researchers, and that in and of itself, those relationships uh, opened doors in so many different directions moving forward. The Hi'ialakai, this was I was on the maiden voyage in 2004. She was recently decommissioned, but we got a lot of science out there, and while we were out there, worked with a lot of MOP students. Um, BZ, Brian Zaglinski, and Joe Laughlin went to school with both of these guys. BZ was actually my team leader at Quest in 2000, and uh, so I was in the same graduating class with these guys as well, all MOP students. These guys are just doing brilliantly. Joe has really taken the aquaculture, actually mariculture road, and he managed Kona Blue Water Farms for a long time, then went off to Panama. He's still in that field. And BZ went on to do his PhD at Scripps and is now doing his postdoc there, where he leads the 100 Island Challenge project in Stuart Sandin's lab. Um, Other fun MOP graduates on the cruises here. This is my second cruise. Paula Ayat and Costa Stamoulis were up there. We were the fish team this year. Paula went on to work on the fish team at NOAA for another decade. And Costa actually found his passion there with fish and started working with Alan Friedlander, did his PhD under him, and really got into mapping the GIS, or Geographic Information Science. So he has just completed his PhD and is working as a postdoc doing the same. Other fun Hilo and MOP graduates working with the Nature Conservancy. This is just an incredible group, an organization, and a lot of fun people. And I wasn't kidding when I said I'm working in the field with everybody I went to school with. Zach Caldwell, Chad Wiggins. Zach is actually now the global dive manager for TNC's diving program. Chad Wiggins is now the Palmyra manager. Cecile and Julia Rose are on the Big Island, and Roxy is on Maui, and all in the Marine program. And then Jen Vanderveer and Erica Perez are with the Coral Reef Alliance. Jen is here on Maui, and Erica's on the Big Island, doing good work for our resources. Two other overachievers, really amazing women here. Jen is actually a how do I put this? She wasn't actually in MOP, but she taught at Quest and has been very active in MOP in her graduate work. She kind of did some of the leading research on invasive algal species in Hawaii. From getting her PhD at UH Manoa, she moved on to NC's, which is pretty much a think tank. It's really, really cool, highly regarded organization, and now and for many years now, she's a professor at Scripps and sends her students out here. Emily Kelly, who you met recently this last week, she was one of Jen's students. And then Lisa Wedding, who I went to school with, is she was a fish person. She also worked under Alan Friedlander for her PhD, and then did her postdoc in Santa Cruz for NOAA, and then moved on to the Stanford Center for Ocean Solutions, and is now a professor at Oxford University. And all pretty amazing, all starting from MOP. So you guys are in a good place. There's a lot you can achieve just from this starting point. And with that, I definitely want to instill in you the fact that if you want to work in this field, you really can. It's absolutely possible. And don't you ever let anybody tell you it's too competitive. And I know you've heard that already. Um, but if you want to do it, just do it. Follow your path, follow your passion. Seriously. Um, just work hard, do good work, show up. 80% of success is showing up. and yeah, do what you love. Just keep doing it. So from here, I'm going to switch to some of the work that I did with the Division of Aquatic Resources here on Maui. We were pretty much looking at the status of Maui reefs. That's what the Division of Aquatic Resources does. Basically looks at the health of the reef, the fish populations over time, trying to understand trends and weed out what factors are influencing those trends. And I know you've seen this chart recently. Tova Calendar, I believe, was showing you this, but I was part of the data collection on this team. Basically, every summer, Division of Aquatic Resources team goes out and collects data in the same exact location at two different depth profiles at every one of these sites, and those are averaged together to give you these data. And so what you see here is data that goes back in some cases as far back as 1993, and you can see these change over time trends. So the red again is significant changes over time. The black is no significant change, and the green is a positive trend over time. But it doesn't tell you why. And in every single location, it has a different basket of why. And but the take-home message looking at this is most of it's red, and that's not good. That's a problem. I'm going to focus a little bit on Kahikili, which I know you've already heard a bit about, but it's a priority site for a reason. It was, it lost 50% of its coral cover in a very short period of time. 10 to 15 years is a very short period of time for a coral. We're seeing lots of corals in distress, lots of nuisance algal blooms, but there's still a lot of complexity that can support fish life. So it's very much worth saving or trying to. One of the insidious problems there is turf algae. So that's that fine filamentous algae. It's really, really small. This is a close-up of a coral head, and what you're seeing here is a change over 2 weeks in February of 2008, and this is a 3-week time period where this section of coral is now dead because the algae has basically crawled over it and smothered it to death. And it does so not just because it crawls over it, but the algae is taking up, algae has a lot of bacterial communities that are taking up the oxygen at the same level where the coral needs it. So that's one of the ways it kills it. And what we see here in this picture is a loss of complexity. And what do I mean by that? The coral heads around there, the pastel yellows, they're tall, they're vertical, right? So um, they have height and spaces in between, and the stuff in the foreground is brown and crumbly and basically is dead, and it's crumbling, and you're losing that three-dimensional complexity, and it's turning into more of a two-dimensional landscape that's not going to house all of the life that uses the coral reef as its home. So and I know you've been hearing a lot about this lately. This is the KHFMA, the Kahikili Herbivore Fisheries Management Area, where basically they put a kapu on taking herbivores, and urchins stop feeding fish just in this area. But the important thing to take home, I guess, two things. One, this is the first of its kind that we know of in the world, where we protected just the herbivores, but more importantly, where the state of Hawaii used fish to turn the tide in reef decline to enhance its resilience. So again, really, really simple concept here, and that is using herbivorous fishes, those fish that are eating the limu, as a means of controlling algal growth on the reef. Now, when we started this project, when I came on, we didn't understand anything about what the fish were eating. Who are the herbivores? Well, we knew who they were, but um, what are they eating? How much are they eating? And um, so there's this huge learning curve. I had a citizen science program where we went out on the reef um, every week, pretty much. We looked at 6 different sites along Leeward Maui, and we did this for 5 years, where we were counting uh, basically bites per minute. What are the fish eating? How much are they eating it? And we came up with some really amazing data that's been used here in the state and across the Pacific. Actually, the NOAA folks were super interested in this data. But one of the really interesting um, data points that came out was this graph. So this is a graph of parrotfish bites um, by size of the fish. So the x-axis has how big of a parrotfish it is in centimeters, so from nose to tail. And on the y-axis is meters square scraped per year. So uh, we came out with this amazing linear relationship that shows that size really does matter. And the bigger fish you have, the more algae is going to clean off the reef. Um, One of the most effective things that we did was what we called site visits. We would invite decision makers out to the reef to show them what was going on. And nothing uh, put somebody in touch with an issue like them seeing it in person. So this is our former mayor, Alan Arakawa, his environmental coordinator, Rob Parsons, and the Coral Reef Alliance crew, our volunteers, and just an amazing group of people. And we go out and show them what's going on. Um, I've taken everybody from the U.S. Coral Reef Task Force to the EPA to uh, folks from all around the country, from Washington, D.C., senators. We've taken them all out and shown them this reef and what's going on. And nothing has been more effective than actually getting them in the water. So successes over the years, we started with the Kahikili Herbivore Fisheries Management Area and TOVA. I talked to you about the watershed management area, the Ridge to Reef initiative. So that's a huge expansion from just our little fisheries management area to an entire watershed. And right here, I'm showing you Lahaina wastewater injection wells, where they are in relation to where the wastewater effluent is coming out on the reef. And this is some of the pivotal work that Megan Daler and others have done over the years. This is a little diagram kind of showing the evolution of management in the area. We started with the Kahikili Herbivore Fisheries Management Area there in red. Shortly thereafter, it was identified by the Hawaii Coral Program and designated a priority site. And then in 2011, it was designated the whole area in red here. The watersheds, Malkatu were identified by the U.S. Coral Reef Task Force as a priority watershed for the Pacific. So what does that really mean? It means instead of spreading the limited dollars around on a lot of different projects, now they wanted to focus all of that money into an area where we could make a solid plan and create a model, if you will, that could be replicated other places in the state. And so that is the Ridge to Reef initiative that Tova came and talked to you about. And these shirts, if you may have seen them around the island, have the best logo ever. If we each do a little, we can all do a lot. So that has been an integral part of my work over the last decade, working with them. But one of the most important studies was this looking at the efficacy of the fisheries management area. So you can't just put one in place and hope it does well. You have to study it and show the science. So we would go out twice a year. We do 80 to 100 transects each time. And we divide it by the different habitat types because there are different classifications out there. And we'd look at fish, we'd look at coral, and we'd look at urchins. And so twice a year for the first 10 years, and we've seen some pretty amazing successes. The parrotfish are up almost 4 times what they were when we started. Surgeonfishes are up 67%. The difference makes sense in the respects that they have different growth rates and longevity. Parrotfish grow a lot faster. They're a larger bodied fish. They live 8 to 20 years depending on species. And surgeonfishes are much smaller bodied fish. They grow slower. They live longer, like 40 to 80 years. So it makes sense that these are different. But the beauty of the fisheries management area is if you're not taking the fish out, then they stay and they grow there. And so this chart, and I know you've saw a little bit about this with Emily, this chart from the top to the bottom is 2008 to 2018, and what you see is that those fish are growing large in the fisheries management area. We start with smaller quantities of smaller sizes, and then over time, you're seeing these larger body sizes come up on the right hand side. You're starting to see an increase in larger bodied fishes over time. And so yeah, we got to watch them all grow up. Also, what you see here in the picture is the bite marks of the corals. Can you see this? Bite marks where the parrotfish have been scraping the algae off of the dead coral, and you can see the white. They're actually scraping all the way down to the calcium carbonate substrate, and that's important because they take away even the smallest spores of the algae, and now new life can come and settle there. And the best part of that is crustose coralline algae, which creates substrate for new corals. Another really, really amazing project that was Megan Ross, one of our PhD studies. She was also a MOP student here on Maui, where she started. Maya, gosh, so many MOP students on this list here. We looked at dead zones, and so in an earlier photo where I talked about complexity and how the rubble is turning it into a two-dimensional substrate. So that's an example of a dead zone, right? So where the reef is just crumbling in. So in order to understand what was happening out on the reef, we went a little overboard and decided to map almost the whole reef. We did a little over 1,100 contiguous 5 by 5 meter cells. That's Megan and I out there doing some assessments. We collected all kinds of different information about the substrate, what kind of corals were there, what kind of dead corals were there, what kind of algae was there, and made a semi-quantitative assessment of each area. And just to give you an example, this photo, hopefully the color on your screen is pretty good. You'll see it's got lots of color, lots of pastels. So that's your live corals. Compared to this picture, which is more dark and brown, and that's because there's lots of dead corals that are covered in algae here. So this is what we would call a dead zone. And this was the resultant map. It was put into a GIS, which is a powerful analytical tool. It takes all of these data and runs statistical analyses on them. And the long story short is red is dead. And remember, again, these are 5 by 5 meter cells. They are not small. You saw Megan and I in one of them, each in one of them. So they're pretty big areas, and notice they're clustered. They're statistically significant clusters, so something's happening in these areas. And so, like all science, it leads to more questions. Another component of the study was looking at individual coral colonies. Each one of these dots represents a transect. And on each of these transects, there are 10 colonies of Parietes lobata, which is lobe coral, and Parietes compressa, which is finger coral. And so 20 colonies on 18 different transects. And from analyzing pictures taken over the course of a year, Donna Brown actually took a bunch of these photos as well. The take home was that 77% of these mortality incidents were associated with this turf algae, this fine filamentous algae that we don't really think of as being a problem, but it is. It's insidious. And it's fueled largely by nutrients. Another really important project as part of the Ridge to Reef initiative was what we lovingly call the Land-Based Sources of Pollution Project, which is a much shorter title than it's actually called. And this is a partnership with NOAA and USGS and DAR and Maui College, where the objective is to look for positive change on the reef as a direct result of mitigation efforts on land. So are the things that we're doing on land to reduce pollution and runoff to the reef, are they working? So we go out several times a year and look at the coral, we look at disease, we look at temperature and sediment, and so that project, we did that for 2 years and then expanded it to the rest of West Maui. So really amazing partnership. Another opportunity was the Maui Coral Reef Recovery Team. So this is a fun story. This evolved from one of my volunteers from that citizen science project I told you about, where we were counting fish bites. One of my volunteers was a PhD who, this gentleman right in the middle with the white beard, Tony Povolitis, he is a professor and loved what we were doing and thought it was really important and we should have our own Maui Coral Reef Task Force. So he wrote a grant. And not only did the grant get funded, but they gave him twice the amount of money and said, do this. So, so what you see here is a collection of some of the brightest minds in all of the agencies and universities around, and cultural practitioners and folks from the county, from the planning department, all got together to actually help Maui and identify problems and make forward steps. So the Maui Core Recovery Team was a product of that. Oh, I have two pictures of us. This went on for several years, and a lot of really amazing things have come from it as well. And the Maui Nui Marine Resource Council hosted this whole effort. Another amazing thing as a product of the West Maui Ridge to Reef effort is the West Maui Kumuai group, which is the community component that Liz Foote has been coordinating over the past few years, and I was on the advisory council. And you guys will have all kinds of opportunities to get involved in these working groups and councils moving forward, and I hope that you do, because the more people that are in their mana'o, the better Maui will be for it. I mentioned earlier that I did some work with coral disease. So Um, the woman here on the top, Dr. Greta Abbey, she was a coral disease uh, researcher at Hawaii Institute of Marine Biology for many, many years, and she was the coordinator for an effort that goes back to 2002, where the U.S. Coral Reef Task Force told all the U.S. jurisdictions that they needed to address certain problems. They need to come up with plans. So she was the coordinator for the climate change and marine disease component of that. And the fruit of that labor was Hawaii's Rapid Response Contingency Plan in coordination with Petra McGowan, who was at the time with DAR and is now at the Nature Conservancy, and Melanie Hutchinson, who is now pretty awesome shark researcher, professor, also a MOP student. It all comes back to MOP. It always does. But so they wrote this, and from this plan, which is still in place today, and it's a living document, came the Eyes of the Reef. So Eyes of the Reef is pretty much the front line, if you will, because nobody knows something's going on out in the water if folks aren't reporting it. So I'll let Donna continue that discussion later, but Eyes of the Reef is something that you definitely want to get involved in. And then I really had a passion for mapping over the years, and as you all know, technology goes really, really fast. So I decided I wanted to do a lot more of this. So I left and went and got some more education on it. It's pretty amazing. This particular study was one I did with Emily Kelly and the 100 Island Challenge here on Maui. So another fun project I was involved in. And then when I came back from that, I got a really incredible opportunity to work with uh, what we call the False Killer Whale Project, which has this very long name, but working with Dr. Robin Baird and the state and Dr. Christy West, who is the stranding. Yeah, that's awesome. So Dr. Christy West was also a MOP student from Maui. I didn't even know that. But she pretty much runs the marine mammal stranding lab at UH Manoa, and is always looking for volunteers and students. Wink, wink, nudge, nudge. Um, she's incredible, uh, but also is and so is Robin Baird, who also needs volunteers. So, just saying. Um, sorry, late on that delivery. But what I'm doing now, which is not on the slide, this is a slide that you should think about a lot. I'm using all of those GIS mapping schools that I went to school for. I'm working at the Pacific Disaster Center here on Maui, and yeah, just doing good work for the planet. But long story short, our world is in trouble. You all know that climate change is a real thing, and but again, we have so many opportunities, and you are the future, and get out there and do some good work for us. Anyway, thank you for your time. I appreciate it so much, and let me know if you have any questions. Donna will give you my email, and don't be shy about it. Okay.
Thank you, Darla. Thank you so much.
All right. Aloha. We'll figure out how to turn this off in a second. Stop share.
Do you want to say any last words?
Last words. Yeah, I'm very excited that you've chosen to take some of the MOP classes. I hope that you'll pursue your MOP certificate. It only leads good places. We have some phenomenal teachers and opportunities here in Hawaii, and there's nothing more important than the resource.
Okay. Thank
All right. Aloha, guys.